Hi everyone, welcome to this episode of Kuiper Labs. In today's video we're going to be discussing how we can predict the genes that you will have, or predicting genes using a tool called a Punnett square. Okay, so just a quick kind of recap or kind of dig into some of the ideas that we're going to need to understand this technique. Okay, so firstly that we recognize that if for almost every one of your cells you have 23 pairs of chromosomes. Okay, so that means one of each pair coming from mum and dad. That means that for every gene that's on those chromosomes, you also have two different versions that we call alleles. So one allele from mum and one allele from dad. Okay, and so we used the example in the previous video thinking about earwax, soft and hard earwax. Okay, so that you'd have a gene for earwax from mum and dad, depending on what genes they had, they'd pass on particular genes to you. There'd be a 50-50 chance of what that gets passed on, so that that will then influence whether you have soft or hard earwax. And so we used that to identify that these genes aren't equal, that the soft earwax gene tends to predominate or be more common than the hard earwax gene. Okay, and the reason for that is that we use these labels of dominant and recessive when thinking about genes. That a dominant gene is one that will be expressed or kind of will give rise to that feature even if there's only one copy there. So you don't need both to have the same copy in order for it to be expressed. Okay, so using this sort of terminology. Okay, so looking at this idea that where you've got two alleles for every gene, so two versions of the same gene, that we introduced this kind of terminology but haven't really come back to it in much detail yet. But we need to kind of develop this vocab before we can move on. And so seeing that those two alleles can be the same as each other, so identical, and so we use the word homozygous, homo referring to same, so and then zygous referring to like the zygote or the that original kind of cell that made you who you are. Okay, so if we think about our earwax genes, that would be two capital W's or two lowercase w's. So these two things are the same as each other. They are homozygous, or they can be different to each other. Okay, so heterozygous, so hetero being different. Okay, so we could have capital W, little w, or we could have little w, capital W. But what we see is that when you've got one of the same, that it doesn't actually, in biological terms, it doesn't actually matter which way around that we do it, that you've got one of each copy. So these are essentially the same thing. So I'm actually you can put an E cross through that if you want to, or you can rub it out. So where we see that biologically that having one of each is only really one combination. Okay, so if we start talking about it's homozygous, this or heterozygous that's just kind of where that terminology comes from. Okay, now one thing that arises when we're looking at this is this idea that that the genes that you have link to the features you have, but don't necessarily that there's kind of some complexity in the middle here. That's saying that you've got particular genes, but you may or may not end up with that feature. Okay, so the genes that you have, the collection of genes that you have for a particular feature, or just even generally, we use the word genotype. Okay, so your genotype, so for the earwax gene might be homozygous, soft earwax, or homozygous capital W. That's so that's the actual letter code of what the genes that you have. Okay, but you and I know that you are not just the sum of the genes that you have particular features that arise. And so we talk, when you think about that, you have the genotype and the features or the traits that you get as a result of those genes, we call your phenotype. Okay, so your phenotype would be soft or hard earwax. Okay, so that's kind of what arises because of the genes that you have, where your genotype might be, this or this or it. Okay, so where we actually look at the letter code that you have. All right, now I'm going to introduce you to the technique that we can use to predict what chance of different genes that offspring will have based on what the parents have, because we're looking at this idea that that the genes that you get as a direct result of what your parents had, you couldn't get them from anybody else. Okay, you don't just kind of catch them like a cold. Okay, And so what we want to do is, so let's say, looking at your original parents and their soft earwax from mum, homozygous kind of soft, and homozygous hard from dad. Okay, now so a Punnett square is actually just kind of like a set of 4 squares that look like this. Okay, and what we're going to do over on the side here and at the top, we're going to write the genes that come from the 2 different parents. Okay, so we're going to talk about mum over here, and we're going to talk about dad over here, up here. Okay, so we put the 2 genes at the top of each column and then on the rows over there. And what we get is that in these 4 boxes, that then we write down the combination of genes that we get by mixing this and that, this and that. So it's a bit like coordinates in geography. So in this box, we will have this one and that one. So capital W, little w. Over here, capital W, little w. Here and here, capital and then lowercase w, and then same thing over here. So just like we did with plotting out our family tree before, where we had it looking like this. Okay, when we went through each of the options, etc., like that. Now we can actually consider it in terms of probability. Okay, that you have a 100% chance, if these were your parents, you'd have a 100% chance of having these genes. Okay, everyone would get the same genes. If you had 6 kids, 10 kids, 100 kids, goodness me, then those would be the genes that you'd have. Okay, so you would have a 100% probability of that combination. Okay, but so now then the reason that this starts to get powerful is, okay, well, it gives us predictive power based on, well, maybe the parents are a bit different. So let's say that so let's say you're the mum and you've got the soft, the heterozygous, so you got 1 of each. And let's say that dad here also has the same. Okay, now I appreciate with the W's it gets a little bit hard to see, but I'm trying to draw them as distinctive as I can. Okay, so what we've got here, so we've got mum, we've got dad, okay, and so then the same as before, that we can kind of start to combine our genes together. So here we've got 2 capital W's, we've got capital and lowercase w, we've got capital and lowercase w, and then here we've got 2 lowercase w's. Okay, so what we would have here is we'd have a 75% probability of soft earwax and 25% probability of hard, because we recognize that if there's 1 capital in the box, then you will have the soft earwax. It's only when you've got 2 lowercase ones that you will get that one. So that means that if you had 20 children, that on balance, you should see this probability. You should see 15 should have soft and 5 should have hard, give or take. Because it still is chance. But it gives us a way to express a probability. Okay, now let's have a look at a third kind of example. Okay, so let's say mum is heterozygous, it's got 1 of each, and then dad is a pure hard earwax. Okay, so has homozygous for the hard earwax gene. Okay, so we can set it up just as before. Okay, so dad is up top, 2 lowercase, mum is 1 uppercase and 1 lowercase. Okay, so now we can go through the same thing as before. Okay, so uppercase, lowercase, and then 2 lowercase here and 2 lowercase here. So now what we see is that you've got a 50% chance of soft and a 50% chance of hard, all because we now have a different combination of genes in the parents. Okay, and so then you can start to predict the probabilities of the different combinations that you have, and that then actually being able to list them out separately. Okay, and so what this does is it gives us predictive power to be able to look at all the possible combinations that might form from those genes and look at generally how common they are likely to be in percentage or probability terms. All right, thanks very much for watching. Bye for now.